Trypanosoma brucei is a species responsible for African trypanosomiasis. In 2009 alone, there were an estimated 30,000 cases of human African trypanosomiasis, or sleeping sickness. This disease was also responsible for losses of about $4.5 billion in livestock productivity per year, with about 3 million cattle deaths. There are numerous problems with current drugs to control this disease, and these include toxicity, poor efficacy, and drug resistance. A vaccine remains the best theoretical approach to controlling this disease. However, there have been numerous problems with this approach. For example, there's been limited funding, hence its status as a neglected disease. But the biggest hurdle is due to the parasite's ability to escape the host's immune response because it's densely coated with a layer of variable surface glycoproteins, and it continuously changes the surface coat. Conventional vaccine development is a tedious process which involves culturing the pathogen in the lab, selecting the antigens, and then screening these antigens through a lengthy process that involves tests with convalescent sera, tests for immunogenicity, purifying and identifying the components. Successful antigens are then tested in animal models before being developed into a vaccine, a lengthy process that can take 5 to 15 years. A new approach, termed reverse vaccinology, is now feasible following the publication of the trypanosome genome. We have identified 3 proteins using bioinformatic tools predicted to be novel surface proteins and with the potential for being novel vaccine candidates. These antigens will be tested in animal models, developed into a vaccine, a shorter process that should take 1 to 2 years. This will be accomplished by achieving the following objectives: expressing proteins that we've identified as potential vaccine candidates using bioinformatic tools, producing rabbit antibodies, and assessing the effect of antibodies on trypanosome growth. The successful candidates will then be tested on mouse models.